As most of you may have learned from this work, the research in this work has been supported by Biodistribution. As most of you have heard during this Congress, and gene therapy holds the promise of a cure for hemophilia, because we can achieve a relatively good clinical benefit with relatively low clotting activity, and we have very good preclinical models which may predict efficacy and safety of our gene therapy. And as you have heard, as we are all very excited by the successful results which have been achieved in adult patients in both hemophilia B and hemophilia A recently by the different groups, showing single intravenous administration of AAV-derived vectors can provide therapeutic levels of factor VIII and factor IX in patients. However, there are still some limitations that need to be overcome, and among all AAV vector gene therapies is currently hardly applicable to pediatric patients, as we have learned from the previous presentation, because AAV vectors do not integrate into cells and may be diluted during liver growth, and the re-administration is challenging, and moreover there is a fraction of patients that is currently excluded from the trials which have pre-existing neutralizing antibodies against the AAV capsid. So lentiviral vectors, on the contrary, are integrating vectors and can potentially be applied to pediatric patients soon after diagnosis. Of course, the way to go there, it's longer, we understand, but that's, in our view, that's where we have to go. We developed a platform for liver-directed gene therapy based on a lentiviral vector, self-inactivating lentiviral vector, which is pseudotyped VSVG, which is not a human virus, and virtually any human has neutralizing antibodies against VSVG, and the vector contains minimal elements derived from HIV for gene transfer, and a transgene expression cassette which stringently targets transgene expression to hepatocyte by an hepatocyte-specific promoter, and the inclusion of the vector of microRNA target sites for a microRNA which is present in hematopoietic lineage cells such as the liver macrophages and the spleen antigen-presenting cells, to abolish any residual transgene expression originating in these cell types, which may trigger immunogenicity. So by this vector design, in the last 10 years in our lab, we have shown that we can achieve stable gene transfer and dose-dependent reconstitution of clotting factor activity in hemophilia models up to 30% of normal in hemophilia B dogs. And this especially by using a hyperfunctional factor IX variant, which I'm sure most of you are aware of, which is the Padua mutation, which increases factor IX activity by 5 to 10-fold. What we observed in our mouse model was a non-linear dose response in which there is an initial phase in which we don't really increase the factor IX output by increasing the vector doses. So we were a little bit more intrigued about this non-linear dose response recently, and actually separated the liver into different cell population and observed that the Kupffer cell, the liver macrophages, are really very active and take up a lot of vector, and reaching up to 20-30 copies of vector per cell, and this sort of plateaus at some doses in which then we reach a higher hepatocyte transduction. So we observed that actually LV are mostly taken up by Kupffer cells upon intravenous administration to peripheral vein administration to mice, and hepatocyte transduction is limited until reaching a threshold dose. So the macrophages act as a barrier for efficient hepatocyte transduction. So how do we modulate phagocytosis? So we have some natural inhibitor of phagocytosis, in particular CD47, which is a known don't eat me signal, which is ubiquitously expressed and interacts with the SIRP-alpha receptor in a species-specific way, and also been shown to be involved in the half-life of nanoparticles and lentiviral particles. So the lentiviral vectors are enveloped vectors, so derived from an enveloped virus, so they receive the membrane from the producer cell during the process of budding. So this allows for the potentially for engineering the composition of the surface of the vector. So we checked whether there was CD47 on the vector, human CD47, because we produce the vector in human cells, 293T. And so cells do have some level of CD47, and also the vector, this is an electron microscopy of a vector particle, has some CD47. But we can increase the level of CD47 by transducing producer cell with a CD47 expressing vector. So we generate vectors which have higher levels of CD47 on the surface, which we call CD47I vectors. And this is a quantification of the electron microscopy. And when we test this vector on macrophages, so we actually observe that while both vector transduce transfer genes at the same extent into control 293T cells, the CD47I vectors are really more protected from uptake by primary human macrophages. On the contrary, we generated cells which lack completely CD47 by disrupting the CD47 gene with the nuclease technology CRISPR-Cas9 and produced vector from CD47 negative cells, which are actually CD47 negative, as you can see here, and now these vectors are more taken up by macrophages. So the levels of CD47 regulate the uptake of the vector in the macrophages. More CD47, less uptake, no CD47, more uptake. So we then tested this in mice, but the problem is that mice don't recognize the human CD47. So we can exploit a mouse strain, which is actually the NOD, which have a SIRP-alpha polymorphism, which can recognize the human CD47. So we delivered the same vector to C57 hemophilia B and NOD mice, and actually observed that there is a tenfold higher factor 9 expression from the NOD strain, which is consistent with the increase in vector copies into hepatocyte, which is higher in the NOD compared to the C57, and a parallel, we had a decrease, a strong decrease in vector copies in Kupffer cells and other antigen-presenting cells in the NOD strain compared to the C57. And when we tested, this was primarily dependent on the recognition of the human CD47 signal, because if we now test CD47 negative vector, in both strains we obtain a similar factor 9 output, similar vector copies in hepatocyte and in Kupffer cells. We then tested the CD47I vector compared to CD47 low parental vector into NOD mice and observed a further decrease in Kupffer cells, in the vector copies in Kupffer cells and dendritic cells, although there was no parallel increase in hepatocyte transduction in this strain. We then also tested, because the macrophages and the dendritic cells are sensor of vector particle and can produce inflammatory cytokines, we tested the innate immune response to CD47I, LV, or CD47-free LV into NOD, and observed that there is consistently with the diminished or increased uptake, we observed that there is a higher rise in pro-inflammatory cytokines and chemokines, which are macrophage related, when using the CD47 free vector compared to the canonical vector, and also the CD47I vector, which is the yellow line in this case, which was the most, let's say, stealth in this respect. So we then generated, we wanted actually to see what happens in vivo. We generated fluorescent virions and performed in vivo to photon microscopy into mice live upon vector administration. The Kupffer cells are marked in red by an anti-FVIII antibody, and the vector you will see in green, and the sinusoids with the blood flow you will see in white. So if you will see in normal C57 mice that most of the macrophages are becoming yellow because the uptake of the vector particles, and this actually happens very soon after vector administration. If we test the vector into NOD mice, where we have the sensing of the human CD47, you will see that there is a delayed uptake of the vector from the macrophages, but at some point, this is a 45 minutes recording, at some point the vector will be uptaken by the macrophages, will be phagocytosed. When we test the CD47I vector into the NOD strain, we observed a delay in uptake, and actually most of the cells remain negative for the vector. You can see here all the macrophages become yellow very soon, and here they will become yellow after a while, and here some of them will actually remain negative for vector particle uptake. So overall, we can conclude that the LV uptake by human primary macrophages is regulated by CD47 levels on LV particles, and the CD47 signal strongly impacts LV biodistribution in vivo because LV phagocytosis is markedly reduced in a mouse strain permissive to the human CD47-SIRP-alpha interaction and results in higher hepatocyte transduction and factor IX expression. And administration of CD47I vector to NOD mice results in decreased uptake by professional phagocytes in vivo and decreased rise in cytokine responses. So we generated vectors which actually have higher levels of CD47 as I showed. We actually generated vectors which also lack the MHC, and I don't have time to go through this data today, but this has been recently published at the end of last year in EMBO Molecular Medicine. So we removed the MHC from the vector, which is a potentially trigger of allogenic immune responses in humans, and so we now have vectors which are MHC-free and CD47I. So we tested now this vector into non-human primates and expressing a codon-optimized human factor IX with the PAD1 mutation, so hyperfunctional factor. And administered by peripheral vein infusion into non-human primates, macaque and emestrina. Three animals have been administered with the control, let's say, MHC-free lentiviral vector, and 3 animals have been administered with CD47I lentiviral vectors at the same vector dose. As you can see, the vector administration was very well tolerated with no increase in, or minor increase, no increase in alanine aminotransferases and a minor increase in aspartate aminotransferase soon after vector administration, and no detectable increase in body temperature and a minor fluctuation, a minor drop in circulating lymphocytes 1 day after vector administration, which may suggest extravasation to the liver and spleen. What we observed was a very high factor IX expression, was actually higher than expected. In the lentiviral vector group, we achieved about 20 to 30% of normal, which resulted in about 100% of normal factor IX specific activity in the animals, which was actually even higher, 2 to 3-fold higher in the CD47I group, reaching up to 300% of human-specific factor IX activity by the CD47I lentiviral vector. When we followed up the animals for 3 months, some of the animals expressing the high levels developed low titer antibodies against which are human-specific, and this also was evident by the appearance of immunocomplexes, and this actually resulted in a artifactually increase in the factor IX, which was detected by the ELISA and activity assay, which are both capture assay. assay, and only in the mice with detectable antibodies, while the mice which were around 100% of normal remain stable for the duration of the study, 3 months after vector administration. So we then performed vector genome biodistribution. As you can see, most of the vector was in the liver and also in part in the spleen, and actually we didn't detect vector into the testis. We did detect some vector, although logs lower, 3 to 4 logs lower, some into the lungs and into the heart, presumably into the endothelium, and also some into circulating T cells, presumably from transduction from the spleen. So overall, in the NHP study, we observed a very mild acute response, a transient lymphopenia, no increase in body temperature, and substantial targeting of liver and spleen by peripheral vein vector administration with vector copies which were 100-fold higher in the liver than in other organs, at least 100-fold higher. And we have shown up to 300% of human-specific factor IX activity and significantly higher factor IX activity by CD47-ILV, suggesting a preferential transduction of hepatocyte at the expenses of a Kupffer cell transduction. And of course, this indicates that we have to go to lower doses to potentially define therapeutic window, which would lead us to about 30% of factor IX activity. And we observed a development of anti-human factor IX antibodies in the highest expressing non-human primates. So overall, what we have seen, we actually observed that there is a more favorable dose response in non-human primates, while in mice we need 2 to 3 E10 TU per kilo to reach about 300% factor IX activity. Presumably, we can go to lower doses than the one that have been currently administered to non-human primates to reach the same, let's say, therapeutic targets. And of course, this is promising and potentially applicable to humans in the future, we hope. So overall, we have generated immune-shielded vector, which are MHC-free and CD47I, which are more resistant to phagocytosis and show improved hepatocyte gene transfer. We have observed a favorable translational profile while moving from mice to non-human primates, presumably thanks to the species-specific recognition of some signal from the virus and some signals which are of human origin into the NHP compared to dogs and mice, as previously shown. So overall, we are confident that lentiviral vectors represent and may complement the reach and broaden the scope of AAV vector gene therapy for hemophilia, potentially in pediatric patients, and potentially for other diseases. So I would like to thank Luigi Naldini, my mentor, and Michela Milani, a great PhD student in the lab who has been involved in the study, and Andrea Noni for the immunology studies in our institute, our collaborators at BioVerative, Rob Peters and Tongyao Liu, the electron microscopy core at the San Raffaele, Molmed for the large-scale vector production, Edward Ayuso in Nantes for the NHP study, and Matteo Iannacone Federico Moalli for the in vivo imaging in the mice. And thank you very much for your attention.